Hey there, thanks for tuning in to Cell and Gene, the podcast. I'm Erin Harris, and you're about to listen to episode 96 featuring my conversation with Faraz Ali. He's the CEO of Tenaya Therapeutics, which is a clinical stage biotech developing therapies for heart diseases. Faraz joined Tenaya as CEO back in 2018, and before that, he was with companies we all know, such as REGENXBIO, Bluebird, and others, and he's currently on the board of directors for the Alliance for Regenerative Medicine. February is American Heart Month, which is a nationwide observance dedicated to raising awareness about heart health and cardiovascular disease. And February is also recognized as Rare Disease Month, with the culmination being Rare Disease Day on the last day of the month. And so the timing of this episode is no accident because, as you'll hear in just a second, Faraz walks us through how the company is developing innovative gene therapies for heart diseases, particularly cardiomyopathies. Tenaya is focused on addressing the underlying causes of heart diseases rather than just treating the symptoms. We also get into critically important topics like pricing and reimbursement, resource allocation, manufacturing, and more. I'm really excited for you for what you learn in this episode. And so without further ado, here is my conversation with Tenaya Therapeutics CEO, Faraz Ali. Enjoy. Hey, Faraz, thanks for joining me on Cell and Gene, the podcast. So happy you're here.
Thanks, Erin, for having me. Looking forward to it.
Absolutely. So tell us a little bit about Tenaya Therapeutics.
Yeah, Tenaya is a clinical stage biotech company based here in South San Francisco. There are a couple of things that make us unique. One is that we're uniquely focused on the heart. 100% of what we do is focused on heart disease. A second thing that makes us somewhat different is that we're addressing both rare and prevalent forms of heart disease. So both rare, severe genetic, but also non-genetic, more prevalent forms. A third thing that makes us somewhat unique is that we're modality agnostic. So while a lot of people know us for the focus on genetic therapies like gene therapy and even now gene editing, but we really follow the science and what is the right way to address the particular form of heart disease that we are pursuing. And so that's why in our early portfolio, we have gene therapy, gene editing, small molecule, cardiac regeneration. I mentioned we are clinical stage. So as we speak, we have 3 programs in the clinic, 2 gene therapies and 1 small molecule. The 2 gene therapies are going after leading genetic causes of cardiomyopathy, genetic cardiomyopathy. Both of them are gene therapy approaches. Both of them have dosed a few patients, and we're beginning to generate data. And one of them, we even shared data towards the end of last year. And the other is a small molecule, which is initially being developed for HFpEF. We completed a phase 1 study a little bit more than a year ago. We are about 100 bright-eyed, bushy-tailed, highly innovative, highly committed individuals, again, mostly based here in South San Francisco. And we have some outstanding supporters and a very wide clinical footprint at this point of about 60 sites in 7 countries working on interventional studies and natural history studies. So TN201, TN401, TN301, those are terms that I'll probably end up using a few times over the course of our conversation today, just describing the different clinical stage programs that we're addressing.
Excellent. Okay. Well, thank you for that detailed description of Tenaya. And next, I want to talk a little bit about, so speaking of certainly heart conditions, what are the key challenges, and opportunities when translating Tenaya's gene therapy platform from, say, these rare genetic disorders to more prevalent heart conditions?
Yeah. So first, I would say that we're doing the right thing by starting with the severe, rare genetic forms of heart disease. One, because that's where the unmet need is the highest. I mean, many of these patients, and I know we'll end up talking about this more later, really severe, really poor options. And so focusing on it there, you've got the right risk-benefit ratio. That's exactly what the regulators generally want to see. So starting in rare, understanding the delivery of genetic material to the heart using our delivery systems, making sure that we're getting the right expression at the right doses, understanding all of that in a rare population is absolutely the right thing to do before we move over to more prevalent forms of heart disease, perfecting the manufacturing platform, et cetera, et cetera. So that's why we started in rare for gene therapy. But you're absolutely correct. Our vision is to eventually make the promise of potentially one-time curative genetic therapies possible, not only for rare genetic conditions, but also for prevalent forms of heart disease. And we already have some programs in our earlier portfolio that are intended to go after more prevalent forms of heart disease like dilated cardiomyopathy or HFrEF or post-MI infarction. So what do we need as a field? This is not unique to Tenaya. I'd say this is true for any gene therapy company trying to make that transition from rare to prevalent. One is we need to be able to do this at lower doses, right? Mm-hmm. So especially when these are IV administered, going and circulating in the entire body, being able to do this at lower doses to avoid the possibility of some toxicities associated with high doses that have been seen in other gene therapy programs. Doing that is pretty critical, and also being able to do this at scale is pretty critical, because the manufacturing scale that works for a small indication won't work for a large indication. So what are we doing to support that? One is we've been innovating with novel capsids and novel promoters so that you can get to the same efficacy at potentially lower doses, which and improve the safety margin. So novel capsids that do a better job of targeting the heart, novel promoters that boost the expression that you get per cell in the heart when you deliver these medicines, also helps bring down the costs. And that also manufacturing, by being able to produce these materials not only at the 500 liter scale, at the 1,000 liter scale, which is where we're operating today, but at 2,000 liter and even 5,000 liter, as has been done for other biologics, that improves the yields, that improves the consistency, it brings down the cost of goods, and makes it possible to offer these medicines at more commercially viable and societally responsible prices that would be necessary to go into larger indications. So those are some of the things I think the whole field needs to work on, and we're already thinking about it. We have novel capsids, novel promoters, and have indeed been working on manufacturing technologies that would allow us to go to larger scales. And that's unusual for a small biotech company of 100 people to have to be solving all those problems, but that's I think that's the cost of the innovation of wanting to go not just for rare, but also prevalent forms of heart disease.
Yeah, sure. And I'm really glad you brought up the manufacturing piece of this, because that's what I want to talk about next. And even given, the size of Tenaya, you, as I understand it, have your in-house manufacturing capabilities, particularly in your genetic medicines manufacturing center. Tell us a little bit about that, and how would you say that in-house manufacturing is specifically contributing to the company's long-term strategy?
Yeah, so it actually starts an excellent question, and we've done what many of our peers have done, which is internalize manufacturing early on. And part of the insight there is that if you flash back a few years ago, capacity was a major constraint. And with anything that's innovative, there's only so many people, only so many CDMOs with only so much capacity available out there. And so internalizing it was partly to solve a capacity problem in the industry, but frankly, more importantly, addressing the technology issues that I mentioned, right? So how do we go from 500 liter to 1,000 to a 2,000 to 5,000 and beyond? That requires actually tinkering with literally the starting materials, the cell lines. That requires you trying to do something and testing it out at smaller scales, shake flask 1 liter, 3 liter, 50 liter, 100 liter, and then see, is that innovation scaling up? Are you getting the titers? Are they improving? Are you getting the same empty to full capsid ratios? So all of that is deep, and it's technical, and it requires a lot of science. And that's not the kind of thing that you can just buy off the shelf from the CDMOs today. And every time you tinker with a different gene of interest or a different promoter or a different capsid, that actually affects the manufacturability and the manufacturing characteristics. So the central insight here is that in medicines that are so innovative, you have to, we believe, internalize the process development and the analytical development capabilities in-house early on. And then that supports then the transition from non-GMP preclinical work to GMP clinical work. And so the real internalization that we did first was the non-GMP capabilities, process development and analytical development. And then the GMMC, as you mentioned, the Genetic Medicines Manufacturing Center, is where we do GMP scale up to the 1,000 liter scale. We've been very successful at that. Both of our 2 gene therapy programs, TN201 and TN401, were all produced in-house at the 1,000 liter scale, excellent product attributes. And I think that success, that allowed us to move quickly into the clinic, no problems with the FDA. And so we're very pleased with that investment that we made. But the real central insight is the internalization of those upstream non-GMP capabilities to have breakthroughs in science of manufacturing, and we have been presenting quite a lot on that at ASGCT last year. We had several posters on small molecule boosters to improve yield in HEK 293 systems. We've done innovations in the SF9 recombinant baculovirus. So we're actually working with multiple platforms and trying to make improvements in multiple platforms, all in service of that vision of allowing this promising science to go from rare to prevalent. We probably didn't have to do all that if we were only focusing on rare, but it's that vision that is forcing us to think deeper about manufacturing science, even at an early stage, so that what we take into the clinic is ready without a lot of modifications to go into those larger indications. Hopefully that answers your question.
It definitely does. And I think, for anyone that I tend to speak to who has an in-house manufacturing component, the biggest thing is that they've made that decision very early, like at the start. And that has helped them, progress forward quickly and efficiently. So you're in good company with that
Yep.
sentiment too. I want to talk a little bit about pricing and reimbursement. And I know when I attend industry events and the topic of pricing and reimbursement comes up, I think that the temperature in the room goes up about 20 degrees because it's I don't call it touchy, but it's a hard topic that we're still grappling with as a sector. And so, given your background in commercialization, how is Tenaya preparing for the potential launch of your therapies, and then, but particularly in context with pricing and reimbursement?
Yeah, excellent question. And I will say it is something we think about a lot because, yes, part of our strategy is to be a fully integrated company. So we've internalized research, we've internalized clinical development, we've internalized manufacturing, and it is our intent to be able to take at least some of the products that we're developing right now to launch ourselves. We know we can't do everything alone, and so there is a very important role for partnerships. For rare genetic conditions where it's a one-time dose, at least some of those programs we'd like to be able to take ourselves, and others we may have to do with partners because that would be the right thing to do, and we can't do it all. So that indeed we do think about commercialization, and some of that thinking does translate all the way to even at the preclinical stage, we're thinking about target product profiles and what must be true for this to be a viable commercial product that is doing the right thing for patients, that fits in the healthcare system, and that can be properly reimbursed. So we do that early thinking. We have a new product planning group. We've done before we ever dosed the first patient in our TN201 gene therapy program, which is going after the leading genetic cause of hypertrophic cardiomyopathy. We had already done global research with physicians, patients, and even payers. So that's an unusual thing to have to do. But again, early stage company, you don't often hear people doing it, but we think it is really important to have that input in your mind before you've even taken things into the clinic, so that you know what needs to be true to be successful at the end. Now, I would say that how do we think specifically of pricing and reimbursement? I've been in this industry for about 25 years, and I cut my teeth early on in my industry experience at Genzyme. And that was one of the sentinel rare disease companies. And you're right, that sort of temperature going up on the topic of price is something that we've seen. Forget about one-time, gene therapies, but back in the day when I was working on chronically administered rare disease therapies, I think the central insights, whether I go back to my time at Genzyme or now, if you are addressing something that is terrible and severe and has a profound impact on the quality of life or the duration of life for individuals, and now you have a therapy that fundamentally changes that curve, right? And particularly in the rare disease space, generally payers have been willing to support that innovation, right? Mm-hmm. Reimbursement has followed. The headlines follow too, but the reimbursement has followed. And the reimbursement has followed as long as the product is doing what it's supposed to do, right? Mm-hmm. It is truly a transformative therapy. That's true whether it's a chronic therapy. I think it's especially true when it is a potential one-time, durable, curative therapy. And so you have seen gene therapies approved from Bluebird Bio, from Sarepta, from BioMarin, from others, and they have gotten reimbursed, right? And in some cases, the prices are uniquely high. They may be 2 million, they may be 3 million per dose. Zolgensma, of course, Novartis has commercialized Zolgensma, an AAV-based gene therapy for spinal muscular atrophy, terrible disease. And they've commercialized that in more than 50 countries, right? Mm-hmm. About more than 4,000 patients have been dosed. So terrible disease, incredible products, reimbursement has followed, even at high prices. And even some of the health technology associations, HTAs, or ICER in the United States, who really critically look at, is this value for money, right? Is this actually cost effective? They look at things like incremental cost effectiveness ratios. And they have actually supported these prices. They've come out and said these are actually responsible prices because of the cost offsets in the healthcare system as a result of this one-time durable approach. So I think we're the value is, as long as the value is there and the products are doing what they're doing and the price has been set responsibly, reimbursement has followed. We can't take it for granted. It is something that we have to think about, and we do think about it. But overall, I think we've been very pleased with what the partnership that we've seen from the payers to pull these forward and to make them available. And in some cases, you might have to do things like risk-sharing agreements or some innovative models to smooth that pathway to adoption. The field has been thinking about it. I've been involved in that as part of the Alliance of Regenerative Medicine, which is an important organization focusing on these kinds of things, among others. We've written white papers about that. I think we've reached a point where the field understands what must be true to guarantee reimbursement, not just in the US, but in other countries. And so I'm hopeful for the future that the headlines may continue to follow, but as long as we're doing the right thing with the science, addressing severe unmet need with a price that has been set at a responsible level that reflects the value, reflects the cost offsets, reflects the improvement in the duration and quality of life, I think the future is bright for Tenaya certainly, and for the entire field of curative, one-time, potentially curative gene and cell therapies.
Sure. It makes perfect sense. I want to talk a little bit about resource allocation, which we talk a lot about on Cell and Gene, especially with sometimes we find, a lot of people under the roof wear a lot of different hats, or, depending on the size of the company or the indications that they're working on, there's just, people are doing a lot of things. And so I would love to know how, as Tenaya advances multiple programs simultaneously, how do you balance resource allocation between the different therapeutic approaches between gene therapy, cellular regeneration, precision medicine? How do you guys do that?
Yes. Every company I've ever been at, it is always a tough balancing act, especially as you make the transition from preclinical to clinical. Then a lot of your resources start to get focused on the clinical stage programs, whether, again, in my past, whether that was traditional modalities or it's a gene therapy, that math doesn't change. So indeed, you have to balance, you have to be relentless in your focus. And that was already true, especially true in the current market environment where resources are more constrained and financings are not always guaranteed. So we, like everybody else, have had to focus our efforts. And I think I have a very clear message on this point, Erin. We are very focused on our clinical stage programs. And so TN201 and TN401, which are active gene therapy programs, those are moving forward. We're actively dosing. We'll talk a little bit later about what we're looking forward to this year. TN301 is another clinical stage program, and that is the small molecule intended for large indications. And part of that effort to balance, we took a pause last year and said, we are going to focus our resources right now on the gene therapy programs and look for, partnering opportunities for the TN301 molecule because it's intended for a large population. Perhaps that's best done with a large biopharma company, and there's been a lot of interest in that one. So part of that prioritization effort, clinical first, within clinical, the rare genetic diseases first, and then the small molecule second. Preclinical work continues in the background, and we're very proud of all the work that we're doing there. We'll be releasing more data on those programs over the course of this year, but those are a clear second priority after the clinical stage programs. And we like all of our science. I think we like to say we can do anything we want. We just can't do it all at once. right? And so focusing on the clinical stage gene therapy program is the right thing to do. And push comes to shove, if there's a choice between at this stage, spending money on, say, the next preclinical program to move into expensive GMP manufacturing and IND enabling studies to bring them to the clinic, versus spending that same money on something that can improve the rate at which we move forward our clinical stage programs. At this stage, given where we are in our history and given where the markets are, we're going to prioritize that spending towards the clinical stage programs and allow the preclinical work to continue in the background, but not pull the trigger on expensive things that will go the fourth and the fifth and the sixth product to come into the clinic, because that's just not sustainable.
Sure. That makes a lot of sense. Before we move on specifically to learn more about Tenaya's approach to treating heart disease, one more kind of broad question for you. What do you see, or what I should say, foresee? What do you foresee impacting the gene therapy landscape in, throughout the rest of 2025, but even in the next handful of years here? Like what real improvements do you see maybe truly coming to fruition?
I think so. I do. I'm part of the. I mentioned earlier. I sit on the board of the Alliance of Regenerative Medicine. So it's an organization that is representing hundreds of innovative gene therapy, cell therapy, gene editing, regenerative medicine, tissue engineering companies around the world. And so I have a broad view of things. And so I think Tenaya is very representative of where things are going. So what are we going to see? First, I think we've seen incredible success, right? We have to take a step back, and sometimes we as an organization have to put it all together. We have seen gene therapies move rapidly through the clinic. We've seen them get approved. We've seen them have transformative impact on patients. I mentioned Zolgensma, which is the first blockbuster curative therapy that we know of, addressing patients in 50 countries. That's incredible. We've done work to show and to quantify how the probability of technical success for gene and cell therapies are significantly higher, 2 to 3 to 4 times higher from phase 1 to approval compared to more traditional modalities, right? That is exciting to be able to now say that we are at this. We have the data to support that these things work, and then the probability of success is higher. So I think part of what we see happening here is more approvals, right? A more rapid, and even the FDA has acknowledged that they expect to see more and more cell therapies approved. So I think the progress we've made will be consolidated and will continue to see more approvals across a range of modalities, both oncology as well as genetic diseases. And then the second thing I think we'll see is that transition to larger indications. It's not maybe it'll happen one day. I think it's here. And a couple of examples I'll point to is my former company, REGENXBIO. They're in a pivotal stage program for wet AMD. That's a prevalent indication that affects the eye. That's not a rare disease. There was recently, AskBio and Bayer are making some interesting progress in a prevalent form of heart disease and congestive heart failure. Very recently, Bayer and their subsidiary BlueRock Therapeutics announced some great data in alignment with the FDA that their phase 1/2 program for Parkinson's can now transition to a phase 3. So that transition to prevalent indications it's not an idea for the future. It's here. It's real. And the impact will be felt across the entire healthcare system. So that's another exciting, I think, evolution that we will see, already beginning to see. And I think there's more behind it. There are people working on diabetes, people working on a lot of other prevalent indications, including us. So I think that's the other thing that's going to happen. Another thing that I'm excited about is actually just the partnership with the FDA. And just they've been very, very supportive of this field. Peter Marks is, of course, a name that many are familiar with, but now also Nicole Verdun. And just the effort to try to help this innovation get to patients faster with various initiatives. And I'll point on one particular example of the regulatory agencies really trying to work with us as an industry to bring the promise of this science forward. Peter Marks is eloquent about talking about platform technologies like gene editing, right? And if you've got a genetic disease for which a single construct isn't enough, you need to have multiple constructs to really address that entirety of the disease. But it's the same delivery vehicle. It's the same, say, gene editing machinery or genome engineering machinery. And the only thing that is different is the guide, right? Well, gosh, there should be an efficient way that if you have 2, 3, 4, 5 of these or more that you need to address the totality of the patient population, there's got to be some efficiencies in how you develop it preclinically, what your IND enabling studies look like, what your GMP manufacturing requirements look like, and what that clinical design looks like. Because it won't work if you have to treat each of these as de novo individual products. And, there has been real thinking on this, in closed doors and open forums about this idea. And that's super exciting, right? That we have made such tremendous progress from a few years ago where the whole narrative about gene therapies was maybe about clinical holds and safety events. And unfortunately, some really bad things that have happened to some patients as we were still understanding these technologies. Flash forward to where we are today, the FDA is absolutely working in partnership with us to help accelerate this exciting science. So I think we're going to see more of that in the future. So as I said, more approvals, more data that confirms this stuff works, more transition into prevalent indications, and I think more creative partnership with the regulatory agencies on how to bring the promise of this science even faster to more patients in need.
Sure. Well, we sure do like that. That's good. I want to talk specifically about Tenaya and your approach to treating heart diseases. So, how would you say, though, that how your approach to treating heart diseases differs from traditional pharmaceutical interventions, and what potential advantages do you see in your therapies?
Yeah, I think it's a good question. And I'd like to start with just thinking about our lead gene therapy program. So TN-201, we are addressing with TN-201 the leading genetic cause of hypertrophic cardiomyopathy. This involves a thickening of the heart, fibrosing of the heart, arrhythmia, and eventually patients progressing to heart failure, transplantation, sudden cardiac arrest. I mean, it's a bad disease. There's about maybe 600,000 patients in the United States alone with hypertrophic cardiomyopathy. And this mutation that we're addressing called MYBPC3, leading genetic cause, accounts for about 20% of the patients. So about 120,000 patients in the US alone. Now, to really paint the picture of what we're trying to do here, this is now in the public domain. We released early and promising data from our gene therapy study in December of last year. The very first patient who came forward was a 27-year-old woman, self-referred from another country, can't name it because we want to protect the privacy and the identity of these subjects. Mm-hmm. She had, by the age of 27, failed the entire stack of the best available medications available to her, approved medications to date. She had received an ICD. She had undergone a open heart surgery to physically remove, debulk, cut out some of the thickened heart that was obstructing her outflow tract, which is preventing from the blood, efficiently being pumped out of the heart to the rest of the body. That's everything. The best that we have available in our healthcare system today had been thrown at her, and she was still had an enlarged heart. She still had New York Heart Class 2 or 3, and she was clearly suffering in terms of her quality of life. That is what came to our doorstep. And then that is the patient that we dosed in our first, she was the first ever patient dosed with TN-201 in the world. We know that there are other patients like that, patients 2 and 3. We also know that there's children with this condition. There are some children who have this mutation that are so severe that they require heart transplant within the first decade of life. Some of them require a heart transplant or will die within the first months or year of life because they're so severe. Traditional modalities, traditional medications aren't going to address the underlying genetic cause in the way that TN-201 is trying to do by adding a healthy copy, the full-length gene, producing the full-length protein, restoring protein levels where they're missing, and that is driving all this terrible disease that we're talking about. So I think that's the key thing that we're trying to do differently. Traditional modalities are not addressing the underlying cause. They're chronic, and even despite us throwing the kitchen sink, in some cases, these patients, they're still progressing. And that's what we're trying to do that's different. And so I think that is differentiated. We certainly, knock on wood, hope that this is going to prove to be meaningful additions to the range of options available to such patients. And we have to generate more data, and we intend to do that, of course, over the course of this year and next. So that's an example of what we think is different. Not only is it one-time, potentially durable approach, but it's addressing the underlying cause, hopefully a better likelihood of response, likelihood of success, and doing something that a traditional modality simply cannot do.
For sure. And ultimately, certainly leading to a better patient journey.
Exactly. Survival, right. Better quality of life, better functional hearts, but able to do things that they weren't able to do before, right? Because many of these patients, I'll give you another example, our TN-401 gene therapy program is going after the leading genetic cause of arrhythmogenic cardiomyopathy due to something called the PKP2 mutation. Again, these patients, unfortunately, for about a quarter of those patients, the first presentation is sudden cardiac arrest and death, meaning you died before you even got the diagnosis. That's a terrible condition. For those who are lucky enough to get diagnosed in time, the intervention is an ICD, right, to prevent that sudden cardiac arrest. But that's not addressing the underlying problem. Their hearts are still progressing towards failure. They're still, instead of getting sudden cardiac arrest, they get these terrible shocks that really affect them, knock them out. And despite that, they still are exercise restricted, meaning that they're told you cannot stretch, you cannot push yourself, because the act of being physically active will actually push your heart so that you would have more of these ventricular arrhythmias that are uncontrolled and require the shocking. So these poor individuals are living in constant fear, diminished quality of life, despite the best available pharmacological therapy and having an ICD. TN-401 can uniquely address the underlying cause, deliver the healthy copy of the gene, same story as 401, right? Produce the missing protein, and hopefully allow them to live better lives. Even maybe their ICDs will stay there, but then they're not living in fear. And eventually, if we can prove it with the data, maybe this will become the frontline, first line standard of care, that you don't even need an ICD, because we've been able to demonstrate that by adding this gene and by adding the missing protein, you now can live a full life and you don't have the risk of sudden cardiac arrest. It may take a while to prove that, but these are the patient stories that keep us going and that lead us to believe that there is something that we're doing here that's unique and special that the current available options can't address.
For sure. And I think certainly that patients and patients' caregivers are, willing to, knowing that there's hope, light at the end of the tunnel, willing to, wait until that day comes, for sure. Yeah. All right, finally, tell us what's on the 2025 roadmap for Tenaya Therapeutics.
Data.
Yep.
And data.
More data.
And dollars. So I mentioned we've really allocated substantially most of our resources towards the two gene therapy programs I just described, TN-201 and TN-401. In the first half of this year, we will generate more data on the first 3 patients dosed in our, what we call the dose cohort 1. And this includes biopsy data as well as clinical and functional data. In the second half this year, we'll start to also have glimpses of biopsy as well as functional clinical data for dose cohort 2, which is the higher dose cohort for TN-201. On TN-401, we dosed the first patient in our study back in November. So the first half this year will be quiet. We'll be collecting biopsies, but in the second half of the year is when we've committed to begin to share some of our early progress, biopsy data, arrhythmia data, hopefully in the second half of this year. In the background, preclinical work continues and other things continue, and we will be making preclinical presentations as well as publications on some of the other work we're doing. Data from our natural history studies that support both of those 2 programs, data on novel capsids and the work that we've done there, again, part of that mission to transition to larger indications, data on new targets that we've identified from our target discovery efforts, data supporting our gene editing efforts in a new program. So there will be a steady drumbeat of data throughout the year, of which the most important will be those clinical data outputs I mentioned, but staying true to who we are, there'll also be preclinical data presented, and the hope will be to continue to raise money. And it's super important for us to continue on our mission, and so that'll be the focus in the 2025.
Remarkable, remarkable. Well, this has been a pleasure for us talking to you about Tenaya Therapeutics and all you have going on and what's to come in 2025 and beyond. And so I thank you sincerely for your time and your insight and for being here with me today. It's been a pleasure.
It's been a pleasure for me. Thank you, Erin.
Absolutely. All right. So that's it for this episode for Cell and Gene, the podcast. Thanks again to Tenaya's Faraz Ali for joining me and for you, our listeners, for tuning in to listening to our discussion today. And we will talk to you soon.